Okay. So sorry about that. We had a call on Jesse's phone. Take 3. Okay, let's see how many we could do this in. Maybe 4? Hopefully 3 is a charm. Okay, pupil. Round opening in the center of the iris. I get spam calls all the time. Like they just plague my phone all the time. So round hole, does it adjust size or is the pupil always the constant diameter? Always adjusting. So the iris and those muscles are always adjusting, opening it up, closing it down. You can shine a light right in your eye, like a little light. We'll do this in physio lab. What happens if we shine a light sort of right at the pupil, right at the iris? It constricts, right? You get pupil constriction. That is to prevent excessive light photons from hitting the back of the retina, okay? So central opening, you've got the sphincter pupillae muscle, okay? Sphincter pupillae muscle, and then the dilator pupillae muscle that actually opens up or dilates the pupil, okay? So these are controlling size of that aperture or that hole, the pupil. And that light reflex that I mentioned, that's a protective response. So the pupil constricts when bright light is flashing the eye. So the iris and those muscles go, the sphincter pupillae muscle clamps down or contracts, and so it eliminates light from zapping the back of the retina. And actually, you are not supposed to look directly into light, right? Not for long periods of time. Even for things like viewing the eclipse, we had that eclipse like 2 years ago during the day. You're not supposed to just stare straight at the sun, right? That's a lot of photons hitting the back of your retina, the back of your eye, and that can not just photo bleach and take them out of commission temporarily, your rods and cones, but it can actually kill the rods and cones in the back of the retina. That means you lose vision in some of those areas of the retina. All right, so the retina, as we alluded to, is the deepest tunic. So it goes, we went from the outer part, we went to the deeper choroid layer and the ciliary body. It's like the middle stuff. Now we're going to the innermost layer, or tunic, right? Layers or tunics. The retina actually has, again, there's a pigmented layer, okay, that has a single thin layer of cute little thin melanocytes that are all spreading along the whole layer, and then a neural layer. What do you think neural, what does that imply? Neural. There's neurons. There's neurons there. It's neural, right? This is where we find the neurons of the back of the eye. The photoreceptors, the bipolar cells, and the ganglion cells. There's also amacrine cells and a few other kind of sideways coming in different types of neurons that aid in the visual response. But the main ones we're going to talk about are photoreceptors, bipolars, and ganglion cells. Photoreceptors are actually, this is the business part of the retina. These are the rods and these are the cones. This is what allows you to see basically kind of black and white, just light and color, different wavelengths of color. So photoreceptors are made of, we have 2 types. We have rods and we have cones. And then the bipolar cells and the ganglion cells help to transmit the visual signals, those wavelengths, the stimuli, to the actual brain. Okay, so if we look at what this looks like, so remember we're in the deepest tunic. We're in the retina that has the thin pigmented layer of melanocytes, and then it's got the neural layer. And the neural layer is actually a whole sandwiched layering of lots of different neurons that are collecting the visual or light information, okay? Here's the cartoon version, which is kind of nice. So if you learn this, it's always easier to make out the real histology or the real anatomy, okay? The microscopic anatomy of the retina, we have, this is the melanocyte or the pigmented layer. They make melanin, so they appear brownish in color, brownish reddish in color. After that, embedded in those sort of melanocyte cells are the rods that look like rods and the cones that look like little carrots, sort of. Hence the name rods and cones. They actually have a cone shape, okay? So then we have the rods and cones. Then we have the bipolar cells that they synapse on. They have neural connections with synapses where they release neurotransmitter, okay? We've got some other horizontal cells in there and some amacrine cells that are kind of layered in there, but the most consistent are rods and cones, bipolar cells, and then ganglion cells. So the third set of layered neurons in the retina in the back of the eye is the ganglion cells. The ganglion cells actually send their process or their axon out, and they all gather up, and they give rise to a nerve that's cranial nerve 2. And do you remember what cranial nerve 2 is? Have you guys learned the cranial nerves, the 12 yet? Got to review as each time we come up and we go through the material. You got to review it afterwards, otherwise it gets to be too much. Cranial nerve 1, 2, 3, 4, up to 12, right? What's cranial nerve 2? Well, take a guess. What are we learning about? Optic. Optic brings in visual information. That visual information gathers by all these axons of the ganglion cells, gathers up, gives rise to one nerve, exits the back of each eyeball, one big fat optic nerve, or cranial nerve 2, okay? So if we look at this, we have the pigmented melanocyte layer, this retina in the back of the eye. The melanocyte pigmented layer, okay? Then we have the outer segments of the rods and cones, which you can see real clearly like this. And then the nuclei of the rods and cones. And then the bipolar cell nuclei. And then the axons of the ganglion cells. Here's the ganglion cells and their axons gathering up and leaving. And they all collect, they give rise, they basically converge into the optic nerve, which is surrounded by connective tissue, and it leaves the back of the eyeball. You can actually, when we're gonna dissect in lab, you'll be able to free up, cut the fat away, and hold the eyeball by the optic nerve. It's a very dense, almost like a cord, that comes directly out of the back of the eyeball. And it's made of these ganglion axon cells. So, something I want to point out, this is the overall anatomy. We'll look at this under the microscope, just like this, and it looks just like this. And we'll dissect it and be able to see the neural layer and the retinal layer. We can actually peel it off the back of the eye. It's very, very thin. You can scrape it and just lift it right up. It's like a thin sheet, all of this. So the pathway of light coming in, basically the vitreous humor, so that gelatinous, it's almost like gelatin that's back here pushing up in the whole sort of posterior segment of the eyeball. That vitreous humor is pushing up on the retina, helping to hold these layers, these neural layers in place. So ocular pressure and enough vitreous humor pushing back helps to maintain the retina where it is and hold everything in place. Notice here at the bottom it says the pathway of light. Light is actually coming in. Okay, so actually we should probably do that. Let's write that down. Because this is one area of anatomy of the eye that confuses people often. So if we look up at the front, we've got cornea, right? And then we've got back here behind this, we've got the lens. We've got a thin layer, the conjunctiva that goes, well, that was nothing. Can you see this on here? Barely. Okay. Well, conjunctiva goes all in front, okay? Then we have that beautiful ciliary body that goes all around the back, right? It goes all circumference around the back. Suspensory ligaments that attach to the lens to hold it in place, right? They're all anchoring it and allowing it to bulge or flatten. All the light has to go through, and actually, if I put this in here, I didn't draw it, let's add it. Let's do a colored one. Let's do a green eyeball, a green iris. Okay, so this is a green iris. It's got the smooth muscle in there. It's got pigment, colored pigment, very interesting. You got the pupil opening, right? Light actually has to go then through, and light behaves weird. It's both waves, different wavelengths. Some are a little more spread out, some are tighter together. That's what differentiates different types of color or different types of wavelengths on the electromagnetic spectrum, X-rays, all different kinds of rays. Light is both waves and photons, like particles. It behaves like both, okay? So it has properties of both. It comes through like a lightning bolt, okay? So it comes through, it has to pass through conjunctiva, cornea, pupil, and actually I didn't say this, but the fluid in here bathing the front of the lens and around the iris is called the aqueous humor. Fluid back here, that big gelatinous gross mass. I mean, I say it's gross, it's pretty interesting. You're gonna see it in lab, up close and personal. Vitreous humor. So the light has to go through all those structures, right? Conjunctiva, cornea, pupil, right there, and then lens, right? And actually this fluid is also penetrating through it. It's a fluid, it's just like water for the most part, saline water. The aqueous humor has to go through that fluid. then it has to go through the lens, then it has to penetrate and keep going through the vitreous humor, that's this gelatinous fluid, gel, and then it gets to the back, it gets to the retina, or the innermost tunic, right? So it had to go through all this stuff. And it bends, light actually refracts or bends as it goes through different components, cornea, water, lens, vitreous or gelatinous humor, and then hits the retina. Now notice the light coming through, what layer, in this neural layer, what layer of cells is it penetrating or going through first, once we hit the retina? Here's pathway of light coming in. I did this to line up with that. So it's going, light has to penetrate through the ganglion cells, through the bipolar cells, through the nuclei and stuff of the rods and cones, and then the light reaches the rods and cones. The photons of light at those different wavelengths finally strike the outer segments of the rods and cones. This means light has to travel or traverse through all those neural layers before getting to the very back ones, which are the rods and cones. Now I ask you, if the rods and cones are the photoreceptors, they respond to the photons and their receptors, specialized complete receptor cells. Why are they back here? Why is this not engineered so these are out here, then the bipolars, then the ganglion cells? Why is the anatomy like this?
Maybe like
Straight on is not healthy. Okay, what else? Does that make sense? That actually is a good way to say it. Straight on, these rods and cones are so sensitive, and they are constantly responding to little photons of light that are penetrating and coming through. If you had them front and center right here, they would probably burn out. Too much overuse, too much overheat, overexposed, and they would just run down and fall apart. They already regenerate their outer segments. They regenerate, they fall off, they get eaten by the melanocytes, and then they regenerate a new one. They do not regenerate the nucleus in the body of the photoreceptor, but they regenerate the outer segment. So this is to protect your very sensitive, very important photoreceptors by having them tucked at the very back of the retina, not front and center. They're like, we're so sensitive, we could face over here. That'll be all a little protection. All we have to get is boop, a photon here, and we'll respond. Photon there, photon there. So it's a protective architecture of the eyeball so that you don't have wear and tear and burnout and overheating from all of that. Okay, this looks like a mess, but yes, okay, so that's why. The retina and the optic nerve. So if we look at where these are located, we've got the neural layer of the retina, the pigmented layer of the retina, so this is all this, and then the choroid layer was the vascular blood vessel layer that goes around it, and then the sclera. So we went over sclera, I didn't mean to do that, sclera, and then the pigmented choroid or vascular layer, and then we have the next innermost layer, the retina, okay? So these actually gather, that's offensive, okay? So these actually, the ganglion cells, their axons leave. Oh my goodness, I just need to select a little teeny one. Okay, the ganglion cells gather up their little axons, they're leaving, and they are long, and they go, and they all exit and leave in this optic nerve. So they're all gathered and leaving, okay? So rods and cones, synapse on, what's the next layer of cells? Bipolar cells, and then those synapse on the ganglion cells, right? And the ganglion neurons send their axons back, gather up, and leave through the optic nerve. This is cranial nerve 2. Yes? And you'll look at this in lab. Okay? We call this whole area right here the optic disc, aka the blind spot. But why blind spot? Why is the optic disc, this disc where the optic nerve issues from, why do we call that a blind spot? Yeah, Jesse.
I remember you did something in high school where, like, if you hold it, there's like 2 X's on a piece of paper, and you hold it up, like one of the X's, you can't see it, but like your mind like fills it in.
Right, exactly right. We're going to do that same thing in physio along with color blindness tests and all these other things and visual acuity. That's right? So we're binocular vision. We have two eyeballs that have overlapping visual fields, and we have a blind spot in each eye. But the other eye and its visual field, incoming information makes up for that blind spot. So, but you have a blind spot, and when you cover one eye, you can figure out where that is. We're going to do the test that Jesse mentioned. So there's a blind spot because right here, there are no photoreceptors, right? There's no rods and cones right here. This is where the information coming from the adjacent areas is gathering up and giving rise to the optic nerve. So there's no photoreceptors. So you literally have a little small patch of a blind spot in the back of the eyeball, of both eyeballs. Okay. Oh, I will back up one thing. In that vascular layer, or the choroid, and all throughout the eyeball, there are blood vessels, arteries, and veins, and capillaries that make sure the eyeball gets enough blood and oxygen and nutrients, okay? So all that is in the eye, and those gather up as well, and they go to and from through the optic nerve. And you'll be able to see that in lab, sort of all these things kind of gather right back up here. You can grab the eyeball right from the optic nerve. It's very, very tough. Okay, quiz. Are you ready for a quiz? It's not really, this is just an interactive activity. Okay, what are these structures? So I want you to look at this for a second. Now you're like, I have no idea. So here's a cough drop looking structure. We're only seeing a little piece of this because the actual eyeball mounted on a slide is big. So, and we'll look at monkey eyeballs because they're a little bit smaller. But if you imagine, you have to get them on a slide. Our eyeballs are huge. So even when you're looking through the microscope, you have to move around to see all the microscopic anatomy of these structures. So this is anterior, I will say that. This is posterior, back over here. Here's the arrow point. It's not, my student took this photo. I think, who took this? Yes, a bunch of my students took great photos several years ago. This is the cough drop structure. This is the lens. So I'll give you that hint. You tell me what the arrow is pointing to. Okay. Ligament, you're saying ligament? Suspensory ligament. Okay, so this is probably part of, this is more than likely part of that whole ciliary body, which is the ciliary processes, the muscle, the smooth muscle. So this whole structure right here is like ciliary body and ciliary processes. It all goes all back here. It's this muscular layer. And you're right though, we'll see it in lab where you see these tiny little fibers connecting it like this. This shot we can't see them as clearly, but in lab you'll be able to focus and go higher magnification. You'll be able to see the little thin hair-like suspensory ligaments. But I would say, yep, suspensory ligaments or ciliary body, all part of that structure right there that anchors the lens and allows it to accommodate or change position. Okay, good. Here's another one. Well, this is kind of a blow up in this sort of, and actually here, can you see there's almost like hair-like little structures that go like this? It's hard to see, but those are the little zonules or suspensory ligaments. What is this structure? It's split and kind of broken. Overall, it's thick. It's all a whole bunch of epithelial cells all packed together in this dense spherical structure. Well, I said it right here, but it's the lens, right? So this is the lens. That's the actual lens, and you'll be able to see that in lab. And then here's another structure. Here we are looking at, here's anterior view, here's posterior back there. What is all this? What? What would you call this? So I said, what is this layered sandwich that's right there? The retina. Retina, right? So we've got sclera back here, choroid or vascular layer, pigmented melanocyte layer, neural layer. The neural layer of the retina actually has the rods, cones, bipolar cells, ganglion cells, so they're all right back there. Okay, photoreceptors, these are the business receptors of the eyeball, okay? Two main types. Rods, very, very sensitive to light. Matter of fact, even in this classroom, more so when you go outside, they are bleached. That means they're inactive. They cannot operate at higher levels of light. They allow vision in dim light, or I call them like black and white vision. So when it's dusk, dawn, at night, you're scrambling around in your house and it's dark, your rods are operating at that point, okay? Your cones operate best in very bright light. These are what enable color vision and high acuity vision. That means high accuracy, basically. They allow you to make out details form in space, right? And they give you color information. So I think of black and white, these are easy, CC, cones, color vision, high acuity color vision. All right, they are neurons, they're not just receptors, right? These are specialized mini little receptor neurons. There are visual pigments in the rods and cones that allow them to perceive the different types of wavelength or pick up on it. Rhodopsin is in rods, opsins are in cones, and the opsins, they're just different types of photopigment or visual pigments. They're interesting because these visual pigments cleave when they're exposed to particular wavelengths of light. And then they trigger a whole signaling pathway inside of the cell that causes it to either fire or not fire, okay? In physio, we go into the whole signaling of that. But for this, rhodopsin is the photopigment in rods, opsins are the ones in cones. They detect green, red, and blue. And if you know art, you know green, red, and blue, those are like the main colors, right? In the color wheel, those are like the primary colors. We can make every other color as long as we have those. We can mix these all together and get brown and black too, right? Okay, so primates are trichromats, and we are a hominid in the primate sort of order, right? So we are humans, hominids, but we're in with this. We are trichromats. Tri-chrome. What does chrome mean? Color. We're tricolor cone individuals, species. So we see green, red, and blue. We have photopigments in the cones for green, red, and blue. All right, so you know what that means. Some other creatures, have you ever heard of dogs being black and white? They can only see black and white? That's a myth, that's not really true. They are dichromats, so they have two. And I believe they have, I think it's green and red, if I'm not mistaken. So they see color, but they don't see the rainbow of color that we see. There's a lot of other organisms that see in different variations because of how many cone opsins they have. So light and heat can damage the photoreceptors. You cannot regenerate them. If you sit there and stare into a UV light or up in the sun for 20 minutes, you will kill many of those rods and cones, and you will not get them back. This is why it can result in blindness. They don't regenerate. They're continuously replacing their outer segments, though. So the outer segments they get eroded, broken down, recycled, and then they regrow them. But you need the soma, or the body and the nucleus of the rods and cones to be able to do that. And if you look closely, I don't know why. If you look closely, you can actually see those distinct layers, right, of the retina. And I want you just to remember, we'll go back to this, where exactly are the photoreceptors in that retina layer? Are they at the front or the anterior part of the retina, or are they at the posterior end of the retina? The posterior end, right? And light goes through in that way. It's to keep them protected because they are so important. They can heat up and get damaged. And you know that, so we kind of went over that. I emphasized that. It makes sense. We should stop here. Okay, so sorry for the choppy video for those of you going to watch this later, but hey, got to just roll with it. Okay, have a great Friday and a great weekend. Do I just hit the red button again?